In this video, we're going to go and have a brief look at photosynthesis and look at how this can be applied to answering exam questions, primarily looking at experiments that look at factors affecting photosynthesis. So, the best way to use this video, in the link below, there is a link for you to go and download a worksheet to go and help to go and fill out during the video. As you're watching the video, go pause it at key points and fill out the answers to the short questions as you're moving through. You can then use the answers to the short questions to help answer the longer questions. So, photosynthesis is the reaction that happens within plants that allow them to convert inorganic carbon dioxide and water into glucose and oxygen. Carbon dioxide comes from the air, the water comes from the ground, the glucose is converted into starch and is stored in the plant, and the oxygen leaves the plant by diffusion. So, in lots of questions, they'll go and ask you about the structure of the plant or the structure of the leaf. It's important that you know the key layers and what each layer does. So, at the top, we have a waxy cuticle. In terms of photosynthesis, the waxy cuticle is transparent, so it allows light through while still stopping water from entering the leaf from above. Below that, we have the palisade layer. So palisade cells have lots of chloroplasts because this is where they're going to go and carry out photosynthesis. Bear in mind that the palisade layer is at the top of the leaf, so this is the section that will probably be getting the most light, or the greatest light intensity. Below this, we have the spongy mesophyll layer. So the spongy mesophyll layer is made out of mesophyll cells, which are rounded and help create large air spaces within the leaf. At the bottom of the large air spaces are guard cells, which allow the stomata to open and close so the leaf can go and control the process of transpiration. Within the leaf, the large air spaces allow carbon dioxide to diffuse in, where they diffuse into the palisade cells, where the photosynthesis is taking place, and oxygen to go and diffuse out, which is how oxygen enters the air. Leaves have veins which comprise of phloem and xylem. So the phloem carry away the glucose, this is made in photosynthesis, and the xylem carry water up from the roots up to the leaves, so the water can then go and enter the palisade cells and be used in photosynthesis. The plant uses the glucose it makes for a number of things, and in many exam questions there might be a short section asking you about the uses of glucose. We could also go and monitor the rate of photosynthesis by going and measuring some of these products. So, the plant can go and store glucose by joining many glucose molecules together to form starch. It can convert it into fats and oils. Using nitrates absorbed from the soil, it can convert the glucose into amino acids. Amino acids can then be joined together to form proteins. And finally, glucose molecules can be joined together in a particular way to go and produce cellulose. Remember that cellulose is what makes up cell walls and helps provide the plant with structure. So, if we now move on to look at our first experiment looking at measuring the rate of photosynthesis. Quite often, photosynthesis experiments will be done using pondweed and putting it underwater. We do this because it's easy to count the bubbles of oxygen coming off it. We'll set it up as shown in the diagram. However, in lots of exam questions, they won't really go and ask about the setup. They'll usually show you some sort of diagram to show this. So some of the general questions that you may be asked is, what is the dependent variable? Remember, the dependent variable is what we're measuring. So in this experiment, we would be measuring the number of bubbles of oxygen coming off of pondweed. What is the independent variable? So the independent variable is what we're changing. So in this experiment, we're changing the distance that the lamp is away from the pondweed. Control variables are things that we need to keep the same each time. So you might want to talk about carbon dioxide concentration in the water or the amount of pondweed. The interval is the distance in between each reading. So usually in photosynthesis experiments, we talk about the interval as being 10 centimeters. Why do we repeat the experiment? So we're repeating the experiment so we can go and take a mean average. We repeat it so we can look for anomalies. We repeat the experiment to show that the results are reliable. Remember that repeating it shows that the results are reliable, not necessarily accurate. Things that could cause errors in this experiment is the fact that the longer you leave the light on, the hotter the water gets. So you might want to consider how you can go and stop the water from getting hotter. So the second experiment, very similar to the first, but instead of counting the bubbles, we go and we put a gas syringe there. Right, a gas syringe will give us more accurate results because it will give us a physical measurement of the amount of gas produced. So, go back, any parts that you want to go rewatch, pause it, go and download the activity in the description below and give it a go.